We are going to begin our discussion on general embryology. Embryology is the development of a human being from fertilization to birth. So we are going to be discussing what happens within the female's reproductive system when you're developing from fertilization to birth. We are going to divide this discussion on general embryology into week 1, what happens in the first week of development. Week 2 shall be looking at what happens in the second week of development. Then we shall look at what happens in the third week of development. Then we shall go and look at what happens between week 3 up to week 8 of development. And then we shall look at what happens between week 9 up to birth. So, those of you who read ahead, you can use this outline and make sure that you read ahead. Reading ahead is very, very important, so make sure that you read ahead. Yesterday we talked about the menstrual cycle, and we looked at one of the phases of the menstrual cycle known as ovulation, when the ovum is released. If that ovum is not fertilized, we shall proceed into the luteal phase, and at the end of the luteal phase we shall have menstruation. But if that ovum is fertilized, a placenta will be formed because there will be an embryo. And that placenta will prevent the corpus luteum from shrinking for about 3 months. And because the corpus luteum will not shrink, the levels of progesterone will remain high, and therefore the endometrium is not going to break down, so you're not going to have menstruation. That is why when you do not menstruate when you're supposed to menstruate, or when you miss your periods, it is likely that there was fertilization, you are pregnant, a placenta has been formed and it is producing a hormone. That hormone is known as human chorionic gonadotropin. So it is producing a hormone which is preventing the corpus luteum from shrinking, so the corpus luteum is remaining intact. It is continuing to produce high levels of progesterone, and that is what has prevented menstruation. So when you get pregnant, you will not menstruate because the levels of progesterone will remain high, and therefore the endometrium will remain intact. So, that ovum which is ovulated must be fertilized by a sperm. And when it is fertilized by a sperm, an embryo is formed, and that marks the first day of intrauterine life. So the first day of intrauterine life is at fertilization. An example here, we are seeing a sperm. These are not many sperms, it is one sperm. We are seeing a sperm penetrating the ovum, and it releases its nucleus into the cytoplasm of the ovum. And when you look at this illustration, there are many sperms which approach the ovum, but only one succeeds. It penetrates the sperm and releases its nucleus into the cytoplasm of the oocyte. On the other hand, the oocyte is ovulated when it is surrounded by a glycoprotein layer known as the zona pellucida. And a layer of cells come with it from the follicle, and this layer of cells forms a layer known as the corona radiata. So the boundary or the barrier around the ovum has its own membrane known as the plasma membrane. It is also surrounded by the glycoprotein, the zona pellucida, and these layers of cells which it came with from the follicle will form a layer known as the corona radiata. So in order for the sperm to enter and release its nucleus inside the cytoplasm of the oocyte, it must pass through the corona radiata, it must pass through the zona pellucida, and also pass through the membrane of the oocyte, and it will release its nucleus on the inside of the oocyte. And that is what we are having here. It releases enzymes from this organelle known as the acrosome inside, and these enzymes digest away the barriers until it releases its nucleus into the cytoplasm of the oocyte. The other thing I did not mention is that this oocyte is ovulated when it is not yet mature. But once the nucleus of the sperm enters its cytoplasm, it completes its maturation, and therefore it is ready for fertilization. The mechanism by which the sperm penetrates the oocyte is known as the acrosome reaction. And before the acrosome reaction takes place, the sperm is surrounded by a layer of glycoprotein. So the sperm, before it penetrates the oocyte, it will first shed off this layer of glycoproteins, as you're seeing. And this process by which the sperm sheds off the layer of glycoprotein surrounding it is known as capacitation. So capacitation is whereby this sperm sheds off the layer of glycoprotein from its body. After capacitation, the sperm begins to penetrate the oocyte, and this penetration is what we call the acrosome reaction. And then it will release its nucleus into the cytoplasm of the oocyte. It means that in the cytoplasm of the oocyte, we are going to have two nuclei. The nucleus for the oocyte, which was originally there, and the nucleus of the sperm, which has just entered. So you have two nuclei. The nucleus of the sperm which has just entered is known as the male pronucleus, and the nucleus of the oocyte is known as the female pronucleus. So at this time, you have two nuclei on the inside of the cytoplasm of the oocyte. These nuclei will then fuse to form one nucleus. These nuclei will then fuse to form one nucleus. And when these two nuclei fuse to form one nucleus, we can say that fertilization has taken place. So fertilization does not take place when the sperm penetrates the oocyte. No. It specifically takes place when the male pronucleus, which is the nucleus of the oocyte, and the female pronucleus, which is the nucleus of the oocyte, when they fuse. When they fuse, they form one nucleus, and that marks fertilization. Fertilization is when the male pronucleus fuses with the female pronucleus to form one nucleus. That is what we call fertilization. And when fertilization occurs, this cell we no longer call it an oocyte, we call it a zygote. When fertilization occurs, we no longer call this cell an oocyte, but it's known as a zygote. And the zygote is formed on day one when fertilization takes place. And it is the first stage of an embryo. The first stage of an embryo is known as a zygote, which is formed at fertilization. So fertilization marks day one of intrauterine life, or fertilization marks day one of embryology. Polyspermy refers to the oocyte being fertilized by more than one sperm. And this is not the normal way things should happen. Under normal circumstances, the oocyte is supposed to be fertilized by one sperm. The parent cells which gave rise to the oocytes and the sperms were known as oogonia and spermatogonia. The oogonia gave rise to ovum, the spermatogonia gave rise to sperm. Those parent cells had a nucleus, and the genetic material in their nuclei was condensed into strand-like structures known as chromosomes. So those parent cells in their nuclei, they had 46 chromosomes. The oogonia had 46 chromosomes, and the spermatogonia had 46 chromosomes. Those cells undergo a process known as meiosis, and meiosis, they divide under meiosis to give rise to daughter cells, and those daughter cells are the sperms and the ova. But during the process of meiosis, their genetic material or their nuclei splits into two, such that instead of their daughters having 46 chromosomes like the parents, for them, they have 23 each. So the sperms have 23 chromosomes in their nuclei, and the oocytes also have 23 chromosomes in their nuclei, but their parents had 46. How did it come to 23? The process of cell division known as meiosis, undergone by reproductive cells, splits that number into half, such that the daughter cells each shares and each will have 23 chromosomes. Why is that so? So at fertilization, the sperms bring in 23 chromosomes, and the oocytes bring in 23 chromosomes. And when fertilization takes place, we get the original number of chromosomes which was in the parents, and that is called a diploid state, which has 46 chromosomes. So the diploid number of chromosomes which was in the parents is restored at fertilization when the sperm brings in 23 chromosomes and the oocyte brings in 23 chromosomes. The diploid number of chromosomes is restored. This is what I talk about here. So at fertilization, the diploid number of chromosomes is restored. At fertilization, also sex is determined. The sex of the embryo is determined. Why? Among the chromosomes in the ova and the sperms, among those 23 chromosomes in the ova and the sperms, there is a chromosome which determines sex, because on it we find the genes which code for proteins which determine the male or female sexual characteristics. So much as the sperms have 23 chromosomes and the oocytes have 23 chromosomes also, one of those 23 chromosomes determines sex. And the chromosomes which determine sex are given letters X and Y. So all oocytes have an X sex chromosome. The X sex chromosome has genes which code for the production of hormones which determine female sexual characteristics. Whereas for the sperms, some of the sperms have an X chromosome and some of the sperms have a Y sex chromosome. So the variety is among the sperms. Because all the ova have an X sex chromosome, of the 23, one of them is X. There is no ovum which has a Y sex chromosome. All of them have X sex chromosomes. But the sperms, some have an X sex chromosome and some have a Y sex chromosome. And the Y sex chromosome contains genes which code for proteins which determine male sexual characteristics. So, if a sperm containing a Y sex chromosome fertilizes an oocyte, that embryo becomes male. And if a sperm containing an X sex chromosome fertilizes an oocyte, that embryo becomes female. That is why here I'm saying that depending on which sperm has fertilized the oocyte, sex will be determined at fertilization. So fertilization has occurred. The first stage of the embryo, known as the zygote, has been formed. It contains a diploid number of chromosomes. It is either male or female, depending on which sperm has fertilized it. And it begins to divide by mitosis. You go and read about mitosis and meiosis. So these three are achieved at fertilization. The diploid number of chromosomes is restored. Sex is determined. Cell division, which is cleavage in embryological terms, cleavage is cell division, is initiated. When I say initiation of cleavage or initiation of cell division, we mean that the zygote begins to divide by mitosis. So these three are achieved at fertilization. And secondly, fertilization marks day number 1 of intrauterine life. Now, this is a simple illustration showing us what happens during the first week of development. When the ovum was ovulated, it was premature. But by the time it is fertilized by the sperm, it would have completed its maturation process. It will be fertilized by the sperm and it will form a zygote. And that zygote is one cell and it is the first stage of the embryo. That zygote is one cell containing 46 chromosomes. It then begins to divide by mitosis. From one cell, it divides to form 2 cells, because under mitosis, 1 cells form 2 cells. And you can see from one cell at fertilization, mitotic division begins to take place, and then we'll have 2 cells. Don't forget that the zona pellucida is still surrounding the entire structure. When the 2 cells are formed, they divide to form 4 cells, still surrounded by the zona pellucida. Then the 4 cells divide to form 8 cells. And as you're seeing that when this division is taking place, the embryo is moving towards the uterine cavity due to the contraction of the uterine tubes. And on the inner lining of the uterine tubes, there is cilia. There is an epithelium which is ciliated. So those cilia beat to and fro and they propel the embryo onwards. From the 4-cell stage, we go to the 8-cell stage, and this 8-cell stage is still surrounded by the corona radiata. And let me talk about the 8-cell stage before we proceed to the next cell stage. The 8-cell stage undergoes a process known as compaction. the cells of the 8-cell stage get compacted. They come closer to each other. And that is what we call compaction. They become tightly bound to each other, or they become compacted, and that is what we call compaction. During compaction, these cells also segregate into inner cells and outer cells. So you are going to have cells of the outer layer and cells of the inner layer. And that process takes place at the 8-cell stage. Compaction, cells become tightly bound together, and then they segregate into 2 layers, an inner cell layer and an outer cell layer. The inner cells are known as the embryoblast, and the outer cells are known as trophoblast. I repeat, the inner cells at which segregate at compaction are known as the embryoblast, and the outer cells are known as trophoblast. So that is what happens at compaction, which begins at the 8-cell stage, which is still surrounded by the zona pellucida. From the 8-cell stage, we go to the 16-cell stage, because each of these 8 cells is going to divide into 2 to form 16 cells, and that is what we call mitotic division. And the 16-cell stage is still surrounded by the zona pellucida. The other name for the 16-cell stage is known as the morula. This morula refers to a fruit known as a mulberry. Now, because this morula looks like that, because the 16-cell stage looks like that fruit known as a mulberry, and the other name for mulberry is morula, that's why the 16-cell stage is known as a morula. So we began at fertilization. The zygote divided into 2 cells, then divided into 4 cells, then divided into 8 cells. 8 cells got compacted, giving us an inner cell layer, the embryoblast, and an outer cell layer, the trophoblast. And then from 8 cells, we went to 16 cells. That 16-cell stage still has the inner layer and the outer layer, and it's also known as a morula because it looks like a mulberry. On day number 4, or day number 5, this morula reaches the uterine cavity. And when it reaches the uterine cavity, it finds it ready to nourish it. The endometrium has enough glycogen to begin nourishing it. So when this 16-cell stage reaches the uterine cavity on day number 4 or day number 5, it continues to divide mitotically and forms 32 cells. From 16 cells, we go to 32 cells. Still surrounded by the zona pellucida. Fluid is going to enter into the 32-cell stage to form a cavity known as a blastocoel. The other name for blastocoel is, I'm going to show it to you. This word is blastocoel. That is the cavity which contains fluid in the 32-cell stage. The other name for that cavity is a blastocyst cavity. So when the 16-cell stage reaches the uterine cavity on day number 4, day number 5, it converts or divides by mitosis. The cells divide by mitosis and they form a 32-cell stage. Fluid enters that 32-cell stage to form a cavity known as a blastocyst cavity or blastocoel. So the entire structure therefore becomes a blastocyst, whereby it is an embryo originally of 32 cells, having an outer layer known as the trophoblast and an inner layer known as the embryoblast, which you obtain that compaction. This is the inner layer known as the embryoblast. This is the outer layer known as the trophoblast. And this is now the fluid which has entered into it when it reaches the uterine cavity. This is the blastocyst cavity or the blastocoel. So the entire structure is known as a blastocyst. and it is still surrounded by the zona pellucida, and it's known as the early blastocyst because the early blastocyst is the one surrounded by the zona pellucida. Then, as development progresses, the zona pellucida breaks off. The zona pellucida breaks off. So the zona pellucida is now going to break off. And when the zona pellucida breaks off, we call this a late blastocyst. And after the zona pellucida has broken off, this blastocyst begins to burrow into the endometrium on day number 6 or day number 7. And the burrowing into the endometrium is what we call implantation. So fertilization marked the beginning of the first week or beginning of gestation. So day 1 was on fertilization, and day 6 or 7 is when we conclude our first week, which ends with implantation. So these are the events which take place in the first week of intrauterine life. And some women, by the end of the first week, they will not even know that they are pregnant. But the process has taken place. And some women only know they are pregnant when they have missed their periods. Because when this blastocyst gets in contact with the endometrium, they form a placenta. This placenta is going to secrete a hormone known as human chorionic gonadotropin, or HCG. HCG is going to prevent the copper sodium from shrinking, just like in the menstrual cycle, but this side it prevents it from shrinking, so that the copper sodium remains intact and continues to produce progesterone. That's why the endometrium does not shed off, because it is waiting for an implantation. That is why when you're pregnant, you miss your periods. So week 1 begins at fertilization and ends at implantation. How does the blastocyst accumulate with water? The outer cell mass, which is the trophoblast, is going to pump sodium inside the embryo. That sodium which it pumps inside the embryo is going to attract chloride ions, so that you're going to have a concentration of sodium chloride on the inside of the embryo. That sodium chloride, which is accumulated on the inside of the embryo, is going to increase the osmotic pressure of the embryo, and it's going to attract water from the external environment onto the inside of the embryo by osmosis, and that water is the one which is going to create the blastocyst cavity. Why does the zona pellucida, what causes it to break off? The cells of the outer cell layer secrete enzymes. Those enzymes are going to digest away the zona pellucida and also enable the blastocyst to implant into the endometrium. The cells of the outer cell layer secrete enzymes which break down that zona pellucida, and they also enable the blastocyst to begin burrowing or implanting into the endometrium. How does the embryo move to the uterus? We saw the embryo moving through the uterine tubes towards the uterus. One of the mechanisms is that the uterine tube contracts, and the contraction of the uterine tubes propels that embryo to move towards the uterus. Number 2, on the inner lining of the uterine tubes, there are cilia, hair-like structures, which beat to and fro, and then they propel it ahead. So those two mechanisms, the contraction of the uterine tube and the propulsion by the cilia on the inner lining of the uterine tubes, propel the embryo towards the uterine tube. Hope donates 23. And the ovum donates 23 chromosomes to form 46. So how many chromosomes does the identical twins have? The cells of the body have 46 chromosomes except the reproductive cells, which have 23 chromosomes. And Bernard Yiki, tomorrow we are going to look at twins, how they form. Twinning is not dependent on how many chromosomes are inside the cell. Twinning is dependent on how many embryos are formed. So tomorrow, as we're having our tutorial, we're going to have a discussion or tutorial on twinning, but twinning is not dependent on how many chromosomes are inside the cells. We are going to see how twinning comes about, but it's going to be a discussion and I'm going to be facilitating it. What causes a man to have one gender of babies only? It is simply because if you have only girls, it means that the X chromosomes are the ones fertilizing. The sperms with the X chromosomes are the ones which are fertilizing. And they have a higher chance of fertilizing the ovum. And if you have boys only, it means that the sperms having a Y chromosomes are the ones which are fertilizing. Which sperm fertilizes the ovum? The mechanisms are not yet known. It is random. It is random. It is random. If a sperm containing a Y chromosome fertilizes an ovum, it becomes a male. If a sperm containing an X chromosome fertilizes an ovum, it becomes a female. What determines that it is random? There is no mechanism which is specific, it's by chance. Albert has a way of determining sex of a child, or it's a matter of chance. It is chance, unless if you have high technology gadgets which can identify a sperm with an X chromosome and you just use manually take it out and fertilize the ovum. But it is by chance. It is there are very many theories which have come out on how you are to have male babies, but all those theories are not experimentally proven, so it is still random by chance. The corona radiata is made up of only cells, so that the sperm just passes in between those cells. The acrosome reaction and its enzymes work on the zona pellucida and the cell's plasma membrane. For the corona radiata, the sperm just swims in between those cells to reach the zona pellucida. So the acrosome reaction begins when the sperm has reached the zona pellucida.